This is part 2 of your cell cycle cell division notes. We're going to start on this side of the big sheet and just take a look, big picture. On the x-axis of this diagram, you have all of the different stages of the cell cycle, right? So after mitosis down here in the bottom left, you have two small daughter cells, and you're going to proceed through G1 growth or gap 1 and get bigger. S for synthesis, you're going to copy your DNA. G2, and then mitosis, and then you're back to your two smaller cells, and then, over and over and over again. So this is essentially time. And we can add that time. We can add that there. On the y-axis is concentration, and we're going to see how these concentrations change and what we're talking about, but let's go ahead and label that concentration. The amount of these different components. So let's scroll down here and see what we're talking about. We are talking about CDK, and don't get too overwhelmed with this. It'll make more sense when we go over it. So CDK is called cyclin-dependent kinase. CDK, cyclin-dependent kinase. So you know it's a kinase, so you know it's going to phosphorylate something because that's what kinases do. But this is dependent on something called cyclin. This is dependent on something called cyclin, right? So remember, kinases, again, they're going to phosphorylate other molecules. They're going to donate that phosphate group, and they're going to activate them, and they're going to make them kind of high energy so that they can do work. So CDK is a kinase that is dependent, though, on cyclin. So over here on this right, I've got this little triangle with this little phosphate group on it. So this is my CDK, my cyclin-dependent kinase. But because it is dependent on cyclin, and cyclin is not near it, it is inactive. Its default state is inactive. It has to be turned on by cyclin, right? And just to remember, when we phosphorylate something, that's going to cause a shape change in whatever protein or enzyme is receiving it, or a lipid, or whatever the other molecule is, and that is going to make it more active. Make more active. It's going to make it ready to do work. All right, here's cyclin. So cyclin is a regulatory protein. Unlike CDK, whose concentration is the same, it's constant, cyclin's concentration is going to vary. It's going to change. It's going to fluctuate. So over here on the right, I've got cyclin. Notice that because these two molecules are going to interact with each other and bond, their shapes match. But this is my cyclin molecule here. And remember that this molecule increases and decreases in concentration, right? So I want you to think of this as a reward for completing tasks. So we're going to make a little note here. As the cell, oh, here's a siren again, I'm sorry. As a cell accomplishes tasks, it accumulates or makes cyclin. So a metaphor for this, or a comparison, if you want to think of it like that, is like a video game, or like Chuck E. Cheese when you were a kid. You go, you accomplish the tasks, you play the games, and you get the tickets, right? And then you can turn the tickets in for something. Or if you're playing a video game and you accomplish tasks, or you collect a number of coins, you go on to the next level, right? So this is kind of similar to how you get points or tokens for doing something in a video game. You complete task or tasks to go on to next level. So you accumulate, you accrue cyclin to move on to the next portion of the cell cycle. Right? That's how this is going to work. And as you accumulate cyclin, it will increasingly bind to CDK. So what you get when they've attached to each other is this cyclin-dependent kinase, CDK cyclin complex, and it's active. It is ready to do work. So this is an active protein, activated, ready to partake in phosphorylating things and kicking the cell into the next phase of the cell cycle, right? So remember that this is ready to phosphorylate. Right? That's what we mean when we say this thing is active. It's ready to phosphorylate and energize other molecules. So let's go up here and see exactly how this works. If you zoom out, or I'm just going to scroll up, what you're going to notice up here is that there's a squiggly line. It's not a straight line. It goes back and forth, and that's because of negative feedback. You're never going to have a straight line anymore on a graph in biology. But this is the concentration of CDK, and it goes all the way across the graph because it's constant. And actually on the other end of this, if you follow the line all the way down, here it is. CDK concentration is high, and it's constant all throughout the cell cycle. And let's just go ahead and draw CDK just so we can remember which molecule we're talking about here. Right, that's the letter P. So there's CDK. And we're going to start at the end of the start of mitosis, actually. So at the start of mitosis, there is no cyclin. There is no cyclin. But as you progress through G1, as you grow, as you synthesize phospholipids and you build the cell membrane and you increase your cytoplasm, you accumulate what's called G1 cyclin. There's a whole bunch of types of cyclin. We don't really have to know them, but we're just going to call this one G1 cyclin. Right? So let's go ahead and make a note. As the cell progresses, it produces G1 cyclin. It's like collecting tokens as it makes its way through the game of G1, right? And then the concentration increases, it increases, blah, blah, all up here to the top. And then you have enough cyclin to phosphorylate, or to activate, to activate your CDK, and your CDK can then phosphorylate everything needed for synthesis. So at the top here, you have a lot of this complex. Remember, this is active. So we can say CDK phosphorylates molecules and enzymes needed for synthesis. And then what happens is cyclin, let me get my little color here, cyclin disintegrates. It breaks down and the concentration of cyclin plummets to 0 again. Right? But notice that the concentration of CDK stays the same. Right? So you've accumulated all your points, you've made all of these complexes that are able to phosphorylate molecules needed for synthesis. They phosphorylate those molecules, and then the concentration of cyclin plummets. And then you repeat. Oops. So I'll say G1 cyclin separates, disassembles. And now you progress through S, right? All the way through S. Remember, there's no checkpoint after S. And then you go through G2 and you accumulate G2 cyclin, and the exact same thing happens over and over and over again. As you copy your DNA and you proofread the DNA and you continue to grow and you make ribosomes and you make proteins, blah, 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 right? You accumulate all of these points, you accumulate cyclin, basically, as you accomplish these cellular tasks, and eventually you get to the point after G2 where you have enough cyclin, you have enough of these CDK cyclin complexes that are ready to phosphorylate anything you need to progress through mitosis, and you've kind of passed the checkpoint. I forgot to mention at the start of this that these dashed lines separate the different phases of the cell cycle. And these big fat arrows, these really nice bolded arrows, are pointing out where the checkpoints are, right? So at the G2 checkpoint, we've maxed out our G2 cyclin, and we phosphorylate everything we need for mitosis, and then just like G1 cyclin disassembles, so does G2 cyclin. So the amount of G2 cyclin plummets. The checkpoint has been passed, and now we can keep going. So now, when all the when we go through prophase and the nuclear membrane disassembles and all the chromosomes migrate to the center of the cell, and they all line up, blah 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 blah. Right? We're accumulating M cyclin. Right? So the story is the same over and over and over again. As you progress through these tasks, you accumulate cyclin that eventually get to a certain concentration endpoint where there's so much cyclin CDK complexes that they phosphorylate anything needed to progress through the cell cycle, and the cell progresses, right? Cyclin disassembles, there it bottoms out down here, and then you can start again. Right, this is what happens over and over and over and over again. So I'm going to make a note here at the top of my G2 checkpoint, just like we did before. CDK is going to phosphorylate molecules and enzymes needed for mitosis. And then you don't have a checkpoint. Oops, there we go. Sorry, there it is. I was like, you don't have a checkpoint. Yes, you do. Here it is. It's right here. There's your mitosis checkpoint, your M checkpoint, where your M cyclin kind of maxes out. And then by that point, once all that's dissociated and you've passed the test, you're back to where you started, back into G1, and you can accumulate G1 cyclin again. Right, and over and over and over and over again, as long as the cell is alive and it does cell division. As an aside, in your book, I think your book refers to something called MPF, mitosis promoting factor. That's simply the combination of M cyclin and cyclin dependent kinase. It's this particular checkpoint right here. It's super. It's not unique or special, but it's a more common location for cells to get frozen up on and stop at. If you remember in 9th grade, when you guys looked at cells that were dividing, mitosis, if you were looking at mitosis, you're looking at prophase, metaphase, anaphase, telophase. The most common phase to see under the microscope was metaphase. And that's because if a cell failed to pass through metaphase, it basically just stopped at metaphase and didn't proceed. So that's why there's a spike in the number of cells that you see in metaphase. It's because they never made it past the M checkpoint, right? They didn't have enough mitosis promoting factor to progress. So fun fact, that's why you saw that. But I do want to give you a specific example of what happens, right? What are some things that happen when active CDK phosphorylates molecules and enzymes? So one of them is right after G2, right before prophase, you know that the nuclear envelope breaks down. So I'm going to draw, let's draw a cell with 3 chromosomes. So they've been copied, they're double-stranded chromosomes, you've already gone through synthesis, and basically this CDK cyclin complex is going to phosphorylate the nuclear membrane. So all these little dots that I'm drawing are phosphate groups. And remember that when you're phosphorylated, you're energized. It's a full negative charge that kind of, it increases your energy and you're jittery and you're kind of moving all over the place. And these little phospholipids can't handle that. So it's basically like little teeny bombs that blow up the phospholipid membrane around the nucleus. And this is exactly how the nuclear membrane starts to break down in prophase. It's not this kind of magic thing that happens. It's just phosphorylation of the phospholipids. So we can say that, we don't have to actually annotate this at all. We can just say that this was the cell membrane, or sorry, the nuclear membrane. The nuclear membrane, and this outer membrane here was the cell membrane. Another way that this works, if we go back to the M checkpoint, one result of getting phosphorylated is the microtubules get phosphorylated by the mitosis promoting factor, the M cyclin and CDK complex. And that causes the microtubules to start pulling the sister chromatids apart. That's dependent on this complex phosphorylating all those microtubules. So those are some real life examples of what is actually happening when these molecules are phosphorylating enzymes or other things that are needed to progress through the cell cycle. If at any point a cell fails to accumulate enough cyclin or enough CDK cyclin complexes, you guys already know what happens. The cell undergoes apoptosis. Right? If you don't make enough, you don't meet the checkpoint, the cell undergoes apoptosis. So pre-programmed cell death. So apoptosis if cell fails to pass checkpoint. All right, let's finish the back of the big sheet. Here's a little bit more detail on apoptosis, and one of the key proteins involved in this is something called P53. And basically, if you have a good copy that you inherited from your parents of P53, you're much, much, much less likely to get cancer than if you have a poorer copy of P53. This is one of the main reasons that people get breast cancer. There are other proteins and enzymes, but this is like one of the biggest offenders for breast cancer and cancer in general. All right, so this in particular, I'm going to go ahead and annotate, is a proofreading protein. And so here's P53. If you don't have any kind of DNA damage in the cell after synthesis or whatever, P53 is unneeded. It doesn't need to hang around when there's no proofreading to do because everything is accurate. So P53 in a normal situation breaks down, and this is good. I'm gonna draw a little happy face. The cell is happy. It's good to go. There's no damage, and P53 is not needed. If there is DNA damage, P53 gets phosphorylated, which means it gets activated, and that causes it to bind to DNA. It recruits other proteins to help repair the DNA, and then the cell can divide. That's another happy cell, right? However, if P53 binds to DNA and it recruits these other proteins, but they are unable to repair the DNA, so DNA cannot be repaired, P53 induces or signals or causes apoptosis, right? This is how apoptosis happens, via P53. And at this point, the cell says, okay, I'm not good enough. I didn't pass my checkpoints. I need to break down. So the cell doesn't just explode. Those are good resources that other adjacent cells can use. So the cell starts to it makes what are called blebs. It basically just bubbles off itself, and these little blebs form and get endocytosed by adjacent cells. So adjacent cells can take ribosomes. They can take little chunks and segments of cell membrane and incorporate them into themselves. It's a way for the cell to get recycled if it doesn't pass the checkpoints, any of the checkpoints. So this is kind of like sacrificing yourself, but allowing the community of your cells to gain the resources that used to make up you. So this is kind of a nice symbiosis between adjacent cells when one cell is leaving the world. But that's the end of your notes. I hope this helped. We'll talk about this more in detail in class. We'll practice some questions, and I will see you all next time.